Hello and welcome back to Microbiology. Today we're going to be discussing things like microbial genetics. In this chapter we're going to do several topics on genetics, but the first thing we're going to have to do is point out what genes are. Genes are what's actually passed from generation to generation, but if you really boil it down, what's actually passed is DNA. So really genetics is the study of DNA. DNA actually is the information that allows us to produce all the proteins necessary for life. But really it also represents as our physical characteristics. So we can look at physical characteristics, we can look at the cell characteristics, the genetic characteristics, or even the actual letter code whenever we study genetics. So whenever we use the word genome, we're actually talking about all genetic material within a cell. Most of this is going to be chromosomal, and a good chunk of this is again DNA, but there are other pieces of DNA out there like the plasmids we find in some eukaryotes and prokaryotes, and then organelles like mitochondria and chloroplasts also again have their own DNA. Whenever we're talking about genomics, we're talking about the study of not just chromosomal DNA but the entire genome. Every bit of what is there. So you can see here where we could find genetic material in things like viruses, eukaryotes, and then bacteria. So viruses again have genetic material in their core inside the capsid. Eukaryotes can have things like chromosomes in the nucleus, plasmids in the outside, things in like fungi and protozoa, and then the literal DNA in the mitochondria and chloroplast. Bacteria, on the other hand, have a single large chromosome and again plasmids, and that's where we find DNA. So the chromosomes are going to be important for us to discuss because chromosomes are actually going to be tightly packaged DNA. In bacteria, again, we talked about having a single chromosome that's usually a circle and double-stranded. I do want to point out that in eukaryotes, most of them have multiple chromosomes that are linear. It's also important to note that most of the time we have, again, paired chromosomes in eukaryotes, meaning that we get one chromosome from our mom, one from our dad. Again, they cover the same basic genes, but we often get blending of those features, and that's why we all should be different. So DNA is the information, but really the information is broken into chunks that we call genes. Genes are the basic packets of information necessary to typically make a single protein. In prokaryotes, it's not uncommon to see a gene actually cover more than a single protein, and as a result, again, just in general, you should know that genes equal proteins. We discussed this idea again, the genes tell us the information necessary to make proteins. So we say the actual information, the code, is called the genotype. But again, a protein will have a physical manifestation, and we call that physical manifestation the phenotype. So whenever we talk about the genome size, there is quite a bit of variation. E. coli, for instance, has 4,000 genes. It's all wound up in, again, a single circular chromosome. But if we were to unwind it and stretch it out, it'd be around 1 millimeter in length. That's actually doesn't sound too big, except for a bacteria is around 1 micron in length. So it's really 1,000 times longer than the bacterial cell itself. If we were to do the same thing with humans, we would find that in each cell we have over 6 feet worth of genetic material. And really that information houses 23,000 genes and really across 46 chromosomes. So the first major topic we're going to hit here is the structure of DNA. Whenever we look at this, it's important to understand that the basic structure of DNA is something called a nucleotide. A nucleotide is made of 3 parts: a phosphate, a deoxyribose sugar in the case of DNA. What do you think the sugar is for RNA? The answer is ribose. And then the third part here is a nitrogen base. So every single bit of genetic material has phosphate, sugar, and nitrogen base. But again, in DNA, we have the phosphate and then the deoxyribose sugar. The nitrogen base is actually where the information is housed. The order of the nitrogen bases tell us what protein to make. So really all the phosphate and the sugar do is make a backbone. It's important to note though that at one end of this backbone we should see the phosphate is the highest point. We call this the 5' end, as noted here. At the other end we see that the actual highest point would be the sugar. So we recognize the sugar ending point as being the 3' end. So whenever we look at this strand, again we have a 5' end here, and we should end way down here in space in a 3' end. I do want to point out the nitrogen bases come really in 4 major forms for DNA, and they're really purines and pyrimidines. So we have adenine and guanine that will always pair with their opposing pyrimidine. So adenine pairs with thymine and guanine pairs with cytosine. So A pairs with T and then G pairs with C. So whenever we actually look at these structures we just discussed, we get a bigger picture. So let's start off with, again, the backbone. The backbone is going to be made of phosphate and sugar. Phosphate, sugar, phosphate, sugar. But notice that means at one end, again, we're going to start with the phosphate, and at the other end, we're going to end with the sugar. So again, we called phosphate end 5', sugar end 3'. But again, the information is housed in these nitrogen bases, and these nitrogen bases are always paired to an opposite base. So G's paired to C, and T's paired to A, and C's paired to G. And so the idea is we have an opposite strand. So really DNA is double-stranded, one here and one here, but what's also relevant is notice how they go in opposite directions. So if I start with the 5' here, the other strand is going to be the 3' end. We call that process anti-parallel. Again, having two strands that are going opposite is again anti-parallel. So again, one end would be 5', or one strand would be 5' to 3', and then the other strand would naturally be 3 to 5. And really direction matters for the discussion of replication, because again, since DNA is double-stranded and we want to copy both strands, we end up having a weird situation because DNA polymerase is very particular. The way DNA polymerase works is only in one direction. We can only make new DNA 5' to 3', and as a result we can use one strand as a continuous template, but the other strand has to be made in tiny chunks. We call these tiny chunks the Okazaki fragments. Again, because we can only see so much DNA available on the template strand at any given time. So as the replication fork continues to move up, we get to see small pieces on this lagging strand that we can copy into Okazaki fragments. Again, the leading strand just keeps trucking along right behind the replication fork until again we finish replication. I do want to also point out to you the origin. The origin is where we would start replication. And notice our replication here is bidirectional, meaning we'll replicate up but also replicate down. So let's discuss some enzymes before we go much further. In order to do this effectively, you kind of need to know many of these enzymes. Things like helicase that unzips the DNA, primase that adds a primer, DNA polymerase. There are two different types, polymerase 3 and polymerase 1. DNA polymerase 3 is the one that does the bulk addition of nucleotides. DNA polymerase 1, on the other hand, is going to be doing the replacing and repairing of damaged DNA. And so the idea here is DNA polymerase 3 does most of the additions. DNA polymerase 1 cleans up any messes that are there. We also have DNA ligase. DNA ligase sticks DNA to DNA. So it really puts pieces of DNA together. Topoisomerases, I don't really care you know so much. Just understand that they're going to do a job of typically unpackaging DNA. So let's talk about semi-conservative replication, and then hopefully we can bring the whole story together in just a minute. So with semi-conservative replication, each daughter strand should be the opposite of the parent strand they're made from. And the daughter strands will be completely new, but they're again paired with an original or parent strand, which means really we're just, with each new strand, we only have half of the DNA being truly new. And that way we can decrease the likelihood of mutational error. Again, if the original strand is a correct strand, the new strand should be the opposite of it, or mirror it. And so if there are any deviations from being the opposite of the original, we can fix that because we have a template. So let's talk about DNA polymerase III and some of the problems with it. DNA polymerase III is very, very particular. It's what's used to make the new daughter strand, but it can only make or add nucleotides in the 5' to 3' direction, which again creates that situation we saw where we had a replication fork that was moving, but we had two different setups for the replication of the DNA. One strand was made continuously because it was in the right orientation. The other one was made in small chunks because it was in the wrong orientation. The other big thing to point out here is DNA polymerase can't just bind to DNA. It needs something to stick to first. And that thing it needs to stick to is called a primer. But primers are going to be made of RNA. And as a result, we're going to have to literally take and put RNA on our DNA and then start copying the DNA with DNA polymerase III. But then DNA polymerase I later comes in and replaces that primer. We also discussed earlier the replication fork. This is the point where we're going to break the double helix with, again, the enzyme helicase. Naturally, before we can actually add any nucleotides, though, we have to have that primer made of RNA attached. That way DNA polymerase III can attach. And we get the leading and lagging strands based off the orientation of the template or original or parent strand. So the idea is the leading strand can be made continuously as it follows along behind the replication fork. The lagging strand, on the other hand, can only make small pieces of DNA as, again, chunks of DNA are opened up by the replication fork. But those Okazaki fragments or pieces will be stuck together using DNA ligase. So I know that we've been pretty straightforward with everything and just used a lot of unique terms, but let's use this picture to really sum it up. So the first thing I want to draw your attention to is topoisomerases. Topoisomerase is, again, unpackaged genetic material. The next enzyme we should look at is DNA helicase, whose job is to actually, again, separate the DNA strands from one another, creating, again, the replication fork, which we see here. With the leading strand, it's important to remember that as we open up new DNA, DNA polymerase III can keep copying the newly exposed DNA. And that makes, again, a continuous strand, which we call the leading strand. The lagging strand is, again, lagging because instead of the replication fork, or instead of the polymerase moving up into the replication fork, the polymerase has to move away from the replication fork. And so we can only copy from the replication fork till the previous strand, making a small chunk of DNA, again, that we call the Okazaki fragments. But notice, let's go through a process of how this could work. Say I just opened up this piece of DNA on the lagging strand. The first thing I can do before I can attach polymerase III is to add, again, a primer. So the enzyme primase adds the primer in this space. And again, with the direction of the template or original or parent strand here, we would have to work away from the replication fork, meaning that I will copy the DNA from the primer on down until I hit the previous strand. And so again, we end up with those small chunks. But do we want a chunk of RNA in the middle of our DNA genome? The answer is no. And as a result, we need to bring in polymerase I to replace that primer with DNA, and then we naturally stick everything together with DNA ligase, resulting in what would be two continuous strands in the very end. And so again, if we have a single origin on our DNA chromosome for bacteria, notice that we would start at that single point and work two directions. And so we'd literally get two rings peeling off of each other until we finally get, again, two daughter molecules of DNA. The replication of linear DNA is much the same as we just discussed, except for one major difference in eukaryotes is that we have not just one origin or replication start point, but multiples. And we still do bidirectional replication, meaning that we can actually copy a linear chromosome very quickly. The other thing that's pretty different are the topoisomerases that are present, because there are additional levels of folding and packaging of genetic material found just in eukaryotic cells. So now that we've talked about DNA replication, let's shift gears to the actual use of DNA. What is it used for? And hopefully at this point you understand that traditionally we believe DNA is used for information to make proteins, which we call the central dogma. And again, the process of making proteins from DNA includes two steps: transcription and translation. Transcription is going to be using DNA as the template to synthesize messenger RNA. We then can take that messenger RNA and then produce proteins at the ribosomes. We call this act of converting RNA into proteins translation. But really, in the time that we've been studying genetic material more thoroughly, we've discovered that the central dogma doesn't really tell us everything that DNA does. In fact, there's a huge amount of DNA responsible for producing RNAs that actually help regulate DNA and its expression. So there's lots of nuance to whether genes are even expressed. Another important thing to understand is that DNA in eukaryotes may have regions that we previously called junk. Again, there's really no junk DNA. Again, that junk is well known for being kind of a buffer for any type of mutational error, but even today we recognize it's responsible for producing several RNA molecules that are necessary for regulation of genes. So as you can see here, we have a visual representation of turning DNA into RNA through the act of transcription. Okay, so in transcription we turn the DNA information into things like messenger RNA, ribosomal RNA, and transfer RNA. But again, once we have our messenger, our ribosomes, and our transfer RNA, we're ready to do translation. Again, translation is going to be turning the messenger, using the ribosome, into polypeptides, so the messenger and the ribosome here, and it's going to turn it into polypeptide chains, as we see here. Again, that act of turning the messenger into the polypeptide chain is called translation. Naturally, we're going to go more into this, but again, I want to remind you of what the importance of proteins is. We've discussed before its necessity for being things like enzymes that help with metabolic functions, but they're also very important in the overall outcome or phenotype of the cell itself. So there is an extensive new area of study that we've seen a lot of growth in something called proteomics, which is really trying to understand what do the proteins do. So genomics was studying the DNA, proteomics is studying the proteins that are made from the DNA. So really all DNA is a blueprint that gives us functional products later called proteins. So the act of transcription and translation again requires us to do two conversions. We call the first act or conversion transcription, where we go from DNA into typically messenger RNA. But notice we only need one strand of DNA to be the template to make our messenger RNA. And we again use the base pair matching rule, so if I had a G on my DNA, I would put a C on my RNA. And they're going to try to tell you several different names for the DNA. I want you to know that the DNA strand that we'll use to make the RNA, we're going to just call a template. Okay, so our template DNA gives us our messenger RNA. And we'll call the opposite strand of DNA, the one not used to give us the information, the non-template strand. But what's more important is that you understand that messenger RNA, when translated, it takes 3 letters of messenger RNA to tell us a single amino acid. So again, we call the 3 letters of messenger that tell us what amino acid to add a codon. But in DNA, we call the 3 letters that represent the codon a triplet. So as we said before, there are some important pieces that we need to actually conduct the activities that we want to do. Again, we need messenger RNA, transfer RNAs, regulatory RNAs, which are not called rRNAs. This is a typo, still persists from the publisher. Really, rRNA is ribosomal RNA. So let's go through what they do. Notice again they fixed the ribosomal rRNA issue on this slide. So again, ribosomal rRNA. Okay, But again, the messenger is the sequence of information to make the protein. The transfer RNA is what's going to carry the amino acids to help us make the protein. Ribosomal RNA helps make the ribosome, which is the structure that makes the protein. But then there are other things like primers, where we discussed, again, being necessary to start DNA replication. And then we have things like microRNAs and small interfering RNAs and riboswitches. What these are regulatory RNAs. These are things that, again, regulate gene expression. So let's illustrate the two more important forms of RNA here, messenger RNA and transfer RNA. Notice again the messenger RNA is literally the message. It is the letter code that actually tells us what amino acid to bring. But if you recall, we use 3 letters at a time to actually code for a specific amino acid. Again, we call a 3-letter code of messenger RNA a codon. And so whenever we read a codon like AUG, we're going to be requesting a very specific amino acid. And in this case, AUG actually encodes for an amino acid called methionine. So methionine is a necessary amino acid we need. So the next thing I want to point out is what brings the actual amino acid to the ribosome is the transfer RNA. And as you can see, the transfer RNA is this big hairpin looking structure that really has 2 relevant parts. The anticodon, which again should be the opposite of the codon, and then the amino acid transfer site. So again, if this codon calls for the amino acid methionine, what should this transfer RNA be carrying? Again, the answer is methionine. So the way we double check it is we line up the anticodon to the codon to make sure, again, this is the right amino acid to be added. So RNAs are necessary for the production of proteins, but they're a short-term copy. So we don't invest as much energy into producing them as we do DNA. And so there are 2 major differences between DNA and RNA in our situation here. Again, DNA is going to be using the sugar deoxyribose, but instead in RNA we're going to just be using the ribose sugar. It's cheaper and still works. The same idea falls in line whenever we talk about thymine that's found in DNA. We actually replace thymine with uracil whenever we produce RNA. Uracil is pretty similar, but again costs us much less energy to produce. And since RNA is a single-stranded short-term copy, we often want to use the least amount of energy we can to make it. The messenger RNA is again going to be the message or carrying the information necessary to make the protein. And we read that message by again reading 3 letters at a time. We call the 3-letter code a codon. The transfer RNA again is what's going to carry or transfer the amino acids to the ribosome. And naturally, that transfer RNA should have not only a carrying region for the amino acid, but also an anticodon. The idea is as long as we match up the codon and the anticodon, we should have brought the right amino acid. rRNA again helps make us the ribosome. And the ribosome is a very complex structure that allows us for that interaction between the transfer RNA and the messenger RNA. So here's the example of everything required to do, again, translation. Notice we have our messenger RNA. It is bound basically as part of the ribosome proper, which would be large subunit and small subunit. But really, again, we read 3 letters at a time and actually bring in the appropriate transfer RNA carrying the appropriate amino acid. So let's do a recap or summary of everything we've done with transcription and translation. But let's start our process off with transcription. Whenever we talk about making any DNA or RNA, we need to use a polymerase. But since we're making messenger RNA, we need an RNA polymerase. And so the idea is we're going to use a template strand of DNA to actually make the opposing strand of RNA. And so we can see this process happening here. So again, the RNA polymerase started or attached way back here, but then is being used as we move forward at this replication fork to do transcription elongation. And then eventually, as we come closer to the end, we hit a stop point or termination. So as RNA polymerase pulls past, again, the stop point, we stop making our messenger RNA. So whenever we talk about, again, translation, it's important to understand that there are some basic rules. Again, we need to read the messenger RNA in groups of 3. We call those groups of 3 the codon, which tells us which amino acid to add. But I would never expect you to be able to figure out which amino acid it is from nothing. So whenever I ask you to do this, like on the exam, I will always give you a codon chart just like this. So all you really have to know is how to use this. So let's point out how this works. Notice on the left side we have the first base position. On the second side we have this, or on the upper side we have the second base position. And on the right side we have the third base position. So say our 3-letter code is actually UUG. So I look up the first letter, U, look up the second letter, U, so somewhere in this box I need to find the G. So UUG is actually the codon for leucine. So let's do another one. What would be the 3-letter, or what would be the amino acid for the 3 letters A, C? So again, A, C, asparagine. So again, use the 3 letters to tell you which amino acid to be added. We will again do this, and you should definitely look over the attached assignments in the module that can help you practice this, because this will be on the test. Those assignments aren't worth points, but they again are going to help you practice for the exam where this will happen. Some interesting things actually happen whenever we look at the genetic code in that codon chart. And often you'll find that many times there are multiple letter combinations that account for the same amino acid. And that's really something called redundancy. And so the idea is if we make a mutational error or mistake, the likelihood it affects the actual outcome is much lower. The other important thing to note is something called wobble, where really the first 2 nucleotides in many cases is all we need to determine which amino acid to add. Whenever it comes to that 3rd letter, it doesn't really change many things. So naturally, if we mess up the 3rd letter through mutation, it doesn't actually change the amino acid added. Look back at the table before and see if you can recognize these things. So here's a nice visual summary of, again, the whole process. So again, DNA triplets are turned into messenger RNA codons, and then we read the messenger RNA codons and match it with our transfer RNA anticodons, which are carrying the amino acid that is added to the chain. So again, every 3 letters of DNA codes for 3 letters of RNA that again match the transfer RNA that carry the amino acid. So translation is again the thing we're going to spend a little more time discussing here. Again, we need messenger RNA, we need transfer RNA, we need amino acids and ribosomes, and we're going to follow really the same 3 steps we did with transcription, initiation, elongation, and termination. There's a lot of detail to this that I don't need you to know. What I really need you to take home at the end of the day is that for every 3 letters of the messenger, that codes for 1 single amino acid, which is carried by a transfer RNA and attached to the polypeptide chain that grows from the ribosome. So by the time we're late in this game, notice a bunch of the messenger RNA has pulled through, but we've also put on several different amino acids to make the polypeptide chain. Whenever we talk about what the messenger looks like, again, we will always have a start codon, and that start codon will always be AUG. But the amino acid that AUG encodes for is something called methionine. So every single protein starts with methionine. There are multiple stop codons. UAA, UAG, or UGA. These things again represent that we're done making the polypeptide and we're ready to release it for folding. So again, once we've finished making the polypeptide, we need to finish folding it. And so one of the first actions to promote folding is the post-translational modification, where we clip off the methionine. Again, methionine starts every protein, but is typically not necessary for the protein's function. So by clipping it off, we're ready to start folding the protein. So as you can see here, we have an example of a protein assembly line, because we're not feeding the messenger through just a single ribosome, but multiple ribosomes, making again multiple proteins at the same time. This is a real thing that happens as visualized here using an electron microscope. So eukaryotic transcription and translation is very similar to what we just discussed, but there are some differences. So one of the first things I want to point out is there are co-translational translations that happen only in bacteria and archaea, but not eukaryotes. Another difference is quite minor, but in eukaryotes, AUG is for methionine, and prokaryotes, AUG codes for F-methionine or formal methionine. Again, the difference isn't important enough for us in this class, so just know AUG codes for methionine. And the last thing is every single messenger RNA that is produced in eukaryotes codes for one single protein, while in many bacterial situations you'll make a messenger that codes for several different proteins, meaning it contains multiple genes. So let's show you why each eukaryote only encodes for one gene. And it's because really in eukaryotes a major difference is we have stuff called the introns. Introns in the past we called junk DNA, and we said it really didn't do anything. But really what we say today is introns are non-coding regions, meaning that they're not responsible for making the proteins. And so what we have to do is we copy all the exons and introns to make our messenger. But once we've made our messenger RNA, we do post-transcriptional modification and remove any introns, sticking only the exons together, producing a what is now template for the messenger or for the polypeptide chain to be made from. So again, we copy the DNA into messenger RNA, we modify the messenger by cutting out the introns, and then we use the remaining exons to make our polypeptide. And that's where the spliceosome comes in. The spliceosome is actually going to take and help remove the introns by looping them up and cutting them out. And then we can stick together the remaining exons to produce our protein from. So whenever we talk about diversity in viral genetics, we've already done this once, but I'll point out that often we have linear or circular arrangements for the genes of viruses. We have single or double-stranded segments. We have RNA and DNA strands, or even retroviruses. The idea here is viral genetics are quite diverse because viruses aren't alive and don't have to follow the rules that living things do. The next major topic we're going to cover is a unique system only found in bacteria called operons. These are basically sets of genes that are used to produce a specific ultimate product. There are two basic types of operons, inducible and repressible operons. We taught you in the past the difference between these words. Inducible means to turn on, while repressible means to turn off. Keep that in your mind whenever we go through this, because what an inducible operon is one that is turned on when it's needed. And all a repressible operon is a set of genes that are turned off whenever they're not needed. So with inducible operons, these are often catabolic operons, which code for enzymes used to break down certain things. Many times these substances that we would break down aren't readily available. So usually we don't use these genes to make these enzymes to break down these things we really never see. And as a result, we only really turn these genes on whenever we find the substrate that we need to break down. So we induce or turn on the genetic material whenever the substrate is present. And typically the substrate is what's responsible for turning on the genes. Repressible operons are the literal opposite. These are usually anabolic enzymes that are used routinely to make things we need for core function. But just like we've described before, if we already have way too much of something, should we keep making more? So there are times where we have way too much of a product, and as a result, we should turn off the genes responsible for making the enzymes that make that product. So by repressing or turning off the genes, we get a decrease in production. And as a result, again, now we can use our energy more effectively. We're going to go through some examples now, starting off with the lactose operon, or the lac operon. The lac operon is an example of an inducible operon. Lactose is a sugar commonly found in dairy, but many bacteria will never see lactose, and as a result don't routinely make the enzymes for breaking it down. But it would make a lot of sense that if we did find lactose, we should break it down. So let's go through the parts responsible for helping control whether the genes are actually expressed. The first thing I want to draw your attention to is a regulator. This is a set of genes that makes a repressor, and that repressor is used to block the DNA. We also have something called the control locus that has two parts, the promoter and the operator. The promoter is literally where the RNA polymerase binds. The operator is like the on-off switch, where the repressor can bind, blocking the DNA. What goes beyond the operator is the structural locus. These are what we call our genes of interest, or the things responsible for producing the enzymes that we're trying to control. The repressor is allosteric, meaning that it can be used to bind or not bind. It's like a switch. And so the idea here is that whenever lactose is present, it actually can bind to the repressor and change the shape of the repressor. The result is then the turning on of the genes as the repressor pulls away from the genes. So let's look at an example of this. Notice we have a regulator way upstream that's going to be responsible for producing a repressor protein. And again, this repressor protein by default should sit on the operator, blocking the genes of interest. So every time the RNA polymerase tries to bind and copy these genes, it runs into the roadblock that is the repressor. But as we said, we really only need these genes whenever we need to break down lactose. So guess what happens whenever we bring in lactose? Lactose binds to the repressor, causing it to come off. Now there's nothing blocking the promoter and operator, and as such, the RNA polymerase can go copy the genes, making again messenger RNA, and using that messenger to make ultimately enzymes that break down lactose. So what would happen to our supply of lactose? Well, eventually it would run out. So we would pop the lactose off the repressor, and in doing so cause the repressor to turn back to its normal form, which sticks on the operator, preventing any more transcription and translation, turning the genes back off. The repressible operon again is the literal opposite. So in this situation, most of the time the genes are being expressed, but there are times where we need to turn them off because we have way too much of the resource we're trying to produce. So let's use the example here, which is going to be an illustration of the production of something called arginine. Arginine is an amino acid, and we typically need lots of arginine to do our job, but there are times where we have way too much arginine. So in this situation, note the default is there's no repressor bound to the operator. So we're going to use RNA polymerase to make the enzymes that make arginine. But if there's way too much arginine present, the overaccumulation can actually cause a binding to the repressor, causing it to change shape, which allows it to now stick to the operator. Can I now transcribe or translate these genes? The answer is no. And as a result, I don't make any more enzymes that make any more arginine. So what happens to my supply of arginine? Well, it goes down. And as a result, again, my supply of arginine goes down, meaning I need to pull this arginine off to use it, causing the repressor to return to its default state, which is off the genetic material. Now that we've introduced you to operons, we should probably talk about an interesting thing with phase variation. I want to point out that phase variation is basically a pretty simple idea that genes can be turned on and off, and ultimately those changes of what genes are expressed result in physical changes and really manifestations of disease. So phase variation is interesting in that a microbe can behave one certain way at one point, but then drastically change at a very closely related point. And so the idea here is microbes kind of have choice, and their choice is based off of what genes are expressed. So antibiotics we will discuss at a later point, so don't stress on them here. It would make sense that we do have drugs that are intended to stop transcription and translation. If they can't do transcription and translation, then naturally they can't cause any more harm by producing proteins, nor are the cells going to live. So let's talk about DNA recombination events. And before we do, let's point out some important parts. One of the more important things to point out is plasmids. Plasmids were again extra chromosomal DNA that were great at being moved from cell to cell. Recombinant is any organism that actually has a different set of genes than what it would originally have. So let's discuss horizontal gene transfer, which is a way to create new combinations. Horizontal gene transfer actually happens in 3 different ways. One of the things we should talk about, though, before we really talk about a lot of horizontal gene transfer is plasmids, because plasmids are often things that can be moved from cell to cell that allow for transfer of genes. Ultimately, again, these plasmids aren't usually absolutely necessary for life, and as such will often carry useful traits that can be passed on when things are, again, useful. For this class, we're going to talk about 3 different types of gene transfer in bacteria, including conjugation, which is the passing of plasmids through something called a pili or pilus, which we talked about way back in chapter 4. Transformation is going to be the movement of genes, but really the genes would be found in the environment, and a cell that's on the brink of death would pick them up in a last-ditch effort to survive. And then transduction would be the movement of genes via viruses, in which again these would typically have to be pro-style viruses that allow for the insertion of DNA and that allows for then the removal of DNA whenever we make new viruses. So let's go through conjugation. Again, they're going to say sex, but I don't like that term because again sex illustrates a production of a hybrid offspring. In this case, again, conjugation is going to make 2 different bacteria more similar to one another by transferring plasmids through a structure called a pili. So again, they're going to do the whole F plus, F minus stuff. Don't worry about it. What I want you to understand is that we can actually take genetic information like plasmids, copy it, and then pass a copy over to the neighboring cell, now making both cells more similar to one another. Really, where this becomes problematic is in things like resistance plasmids that can be passed from one cell to another, allowing for the passing of antibiotic resistance. This happens a lot in conjugation because cells recognize a need for it and then can offer again assistance to other cells to make them also survive. So where this can go wrong is whenever one cell has resistance and can pass it to another cell, we see this mass passing of resistance, allowing for individuals that are not responding to antibiotics anymore. In transformation, again, we need a donor that actually typically dies, and as a result, its genetic information can be found readily in the environment. But again, the cell that's going to pick up the genetic information has to be what we call competent, which really means that they have holes in their membrane that allows for genetic material to move across in large amounts. And we're going to use that DNA and try to express it in a last-ditch effort for survival. So in order to really do transformation, you need to try to kill a cell after it's been made competent, and in which case then the genetic information will be picked up from the environment and expressed in a hopefully opportunity to save the cell. Transformation was really first discovered in the 1920s by Frederick Griffith, and what he did was he was looking at 2 different versions of strep pneumoniae. Strep pneumoniae, as you can imagine, causes strep, but there were 2 different strains he looked at. An encapsulated strain, or one that had a capsule, that he called the smooth strain, and then a strain that lacks a capsule that he actually called a rough strain. Which would be more dangerous to us? The answer would be smooth, because again, encapsulated bacteria can prevent phagocytosis, which means they're more likely to survive and cause infection. So let's look at what he did. he actually purposely introduced the smooth or encapsulated bacteria into a living mouse, and the result was the death of the mouse. If you don't know, upside down mouse is dead mouse, and again, that shows that this bacteria overcame the immune system and killed. But what happens whenever we put the non-encapsulated or rough bacteria in? The mouse survives, and it's because the mouse immune system can actually break down the bacteria, preventing it from causing infection. But what happens no matter what strain whenever we add heat to something? Whenever we kill bacteria, again, it shouldn't cause infection because it can't increase in numbers. But one of the weird things he noted is whenever he mixed the dead encapsulated bacteria with the living rough or non-encapsulated bacteria and put them both in the mouse at the same time, the result was a dead mouse. And what he could pull out was then both encapsulated and non-encapsulated living bacteria. So how did this happen? Really, what happened was the living non-encapsulated bacteria picked up the genetic information to make capsules, allowing them to produce the capsules that allowed them to survive, reproduce, cause infection, and kill the mouse. The last thing we're going to talk about for horizontal gene transfer is transduction, which requires a virus, like a bacteriophage, that can again infect a host and carry genes to another host. So in generalized transduction, the idea is that we will insert the viral DNA, we'll start making the parts, but a common strategy is to actually start breaking down the DNA of the host cell. And as a result, whenever we start assembling parts, sometimes we accidentally insert into a virus non-viral DNA, in fact DNA from the host. So what happens whenever that virus goes to infect a new cell? Well, it doesn't put in DNA that is viral DNA, it puts in the previous host DNA. Again, transferring DNA from one host to another host. In specialized transduction, we're going to see a different situation. And here we're going to require a provirus or a prophage. So notice in this example that we had a virus infect this cell and insert its DNA into the host DNA, also called lysogenic. Notice that whenever it comes time to induce or cut out the DNA, the host accidentally cut out partial viral and partial host DNA. So all the new viruses that are made are going to have partial host and partial viral DNA. And as a result, whenever they go to infect a new host, they're not just infecting with viral DNA, but also previous host DNA, which again is a transfer of genes from one host to another. So transposable elements are an interesting topic because what transposable elements or TEs do is they literally jump from one location to another. The interesting part of this is it's kind of random in how it happens. And so the idea here is, why would we want genes that randomly jump around? Those would cause mutational changes that could potentially kill. And that's true. But here's the thing, since bacteria don't do sex, they need a new mechanism to create recombination. And transposable elements are that mechanism. Whenever we talk about this, yes, there will be change. But change can either be good, bad, or neutral. But if you stop and think about how really pressures in evolution work, if again a bad gene is created, what should happen to that organism? It should die. And so should its genes be passed forward into future generations? No. What about a neutral gene? Will that gene become more common because it offers a benefit? The answer is no. Again, it's neutral, it didn't change anything, so again, neutral changes don't get really pushed forward in any major way. But what about a change that is beneficial? Those beneficial changes are the ones that will be pushed forward, and those are the ones that again will lead to new generations and versions through history. So really we have insertion elements, which are the smallest TEs, are really two tandem repeats, really trying to break things up. Retrotransposons are a type of TE that can transcribe DNA into RNA and then back into DNA for insertion in a brand new location. So these are physically going to be coming out of the DNA, being turned into RNA, and then turning RNA back into DNA to reinsert into the DNA. And again, the whole idea here is that we could create this genetic shuffling. Because again, those good changes, that good change or shuffle to the genes will be pushed forward. While again, anything that's neutral or bad won't. Again, bad genes die with the host. So now let's apply this idea to something called pathogenicity islands. This isn't an easy thing to think about, but let's try to kind of work through it together. Imagine you have a strip of DNA with a bunch of genes responsible for causing dangerous effects of bacteria. But imagine I have transposable elements on either side. If I inserted randomly the TEs in between the pieces of information responsible for making unique traits that cause disease, I can create potentially a new combination to create a new effect. And so what I'm getting at is by doing this shuffling, we can create different variations even in the same bacteria. And as a result, we get different pathologic features presented. So one of the last things we're going to have to do is discuss mutations, because mutations are very Before we can really understand fully what mutation is, we should probably point out that there should be a base set of genetic material found in most organisms of the same species. And so we refer to that type of genetic material as being wild type, what we would expect if we were to just pull a random organism and see what genes it has. But really, a mutation allows for the formation of a mutant strain. So this, again, version of the species is a strain that has unique differences. And again, if those unique differences are actually beneficial, those genes will become more common. So at some point, mutant strains become wild types. So what causes mutation? Well, things like spontaneous mutations, which are natural and rising from typically errors in DNA replication. But we also see induced mutations that happen as a result of mutagens or compounds that promote mutation. Some of the chemicals we're concerned with include things like nitrous acid, bisulfites, ethidium bromide, nitrogen-based analogs, really anything that can disrupt genetic material. The interesting thing with the nitrogen-based analogs is these things look like nitrogen bases, so what they actually do is compete with the real nitrogen bases to bind to DNA. So we talked about the replication fork and how we only want to open it enough to copy, and it's because we don't want any nitrogen-based analogs to get in there and disrupt the whole system. Another thing people don't think about is radiation agents, like ionizing radiation like gamma rays and X-rays, or even UV radiation. What radiation is going to do is ultimately destroy DNA, either through causing breaks in the structure or literally causing changes in the way the bonds form, like in the UV case. So whenever we talk about mutations, there are going to be several different types. One of the types we should point out is a point mutation that really only affects a single base, so again, a single point along the gene. This can be things like addition of letters that didn't exist, deletion of letters that should have been there, or even substitutions where we put the wrong letter in. Whenever a mutation is bad, it typically leads to death, so we call these types of mutations lethal mutations. A neutral mutation is one that doesn't actually change the outcome, and as a result, really doesn't offer any minuses or pluses. So again, this is something that doesn't really lead to a hell of a lot of change. Missense and nonsense are different though. Missense is whenever we make a change that leads to the placement of a different amino acid. So we had a difference in the protein being made. And as a result, again, that difference can be substantial enough to have physical manifestations. Nonsense is typically never a good thing, where we add or delete or substitute a letter that allows for the premature stop codon to be present. Again, if we stop a protein before it's made completely, we're not going to get the protein product, and that's nonsense. And so the idea is, again, this is typically never good. So notice we have examples of all these types of mutations. As you can see, we have the normal genes represented up here with the DNA and RNA and the appropriate amino acid. But you can start looking through, notice a missense where we turned a G into a T here that results in a different amino acid added. Notice again in this comparison with base substitution where we again replace the G with an A that results in a U in the RNA, but notice we still added the same amino acid. So again, that would be silent, meaning there was no actual change to the outcome or the protein. Notice where we see more problems is with the frame shift. If I add or delete a nucleotide, there is now a different number of letters there, creating a shift in the 3-letter reading window, meaning that every single amino acid from that point on has the potential of being wrong. And as a result, we're not typically making the right protein. So as we said before, silent mutations are going to be ones where there is a change in the letter code, but the outcome of the protein is not at all different. And again, things that buffer this include the redundancy and wobble. Back mutations are interesting because there are times where a gene can be changed or mutated, but then will go through mutation again that brings them back to the original composition. And with this, we're concerned with production of vaccines. There are many vaccines that are produced that we genetically modify the thing so that it's no longer dangerous. But if it undergoes a back mutation, it can actually become dangerous again. Frame shifts we illustrated earlier were again very bad because it changed the reading window. By shifting the reading window either left or right, we now get the wrong 3-letter combinations that we would usually have. And as a result, the outcome is not typical. But good news is there are systems to fix mutations, including things like photoactivation. This is repair of things that are damaged by UV light. Excision repair is another situation where we're going to exercise or cut out the bad portion of DNA and replace it with the correct portion. Mismatch repair again allows for the recognition of mismatched bases. Again, we have the base pair rules where A pairs to T and G pairs to C. So if again we get a mismatched pair, we know that we need to fix and replace that. I don't care a lot about the Ames test in terms of how it functions, but I do care that you understand why we do it. We talked about a little bit ago this idea that chemicals can even promote mutation. And so really what the Ames test is doing is trying to detect chemicals with carcinogenic potential, or the ability to create DNA problems or malformations. And so the idea is we again run a control versus a test to see whether or not we see changes based off of the chemical we introduced. So we kind of introduced this back whenever we started mutations, but there's this idea that mutations happen and often are random, but can be again a leading kind of changing force for evolution. And so it's important to understand that really mutations can be good, bad, or neutral. In the case of neutral, nothing really changes. But again, bad genes should die with the organism and not be passed on to future generations. But good genes should be passed on. And as a result, we see eventual changes that lead to evolution.